Hi everyone, welcome once again. Before we get started, I would like to briefly introduce Academy, as always, which was created with a clear purpose to bring together expertise from the lab, the field, and the public health. We are strong believers in the One Health vision, and it is only right to think that real progress happens when these three perspectives work together. So over the past year, Animalia has also been strengthening its own research department. During this time, we've successfully developed robust and reproducible challenge models for PRRS, Glasserella, and post-weaning diarrhea in piglets. At the same time, we continue expanding our work to other pathogens, building the scientific tools and expertise needed to support vaccine development and the evaluation of new pharmaceutical products. So be sure to follow the next episodes of Academy, where we will explore these projects in greater depth and share insights from our ongoing research journey. It is a pleasure to introduce Dr. Enric Vidal, a leading veterinarian and expert in animal prion disease. He completed his veterinary degree and PhD at the Universitat Autònoma de Barcelona, where his doctoral research focused on the pathogenesis of bovine spongiform encephalopathy. Since 2002, he has led the PRIOCAT lab at IRTA-CReSA. He also manages the slaughterhouse support service from the Catalan Health Department and collaborates widely on animal health research, including tuberculosis, African swine fever, and other emerging diseases. Since October 2025, he has coordinated the scientific committee advising the Catalan government on Lumpy Skin Disease. Although Enric's collaboration with Animalia is recent, Animalia has a long-standing history of working closely with CReSA, making this conversation a continuous of an established and valued relationship, of course. In today's episode, Enric joins us at Academy to talk about lumpy skin disease and share the latest updates and insights on this emerging disease. Hello, Enric, how are you doing?
Yes. Good morning, Ariadna, and everyone. First of all, I would like to thank Animalia and yourself for inviting me. It is a pleasure to be here this morning. And now that I have your attention, and you've mentioned my participation in certain studies in CReSA with African swine fever, I would like to take the opportunity to remind the audience that the studies of genome sequencing have finally demonstrated that the virus of African swine fever circulating in the area where our laboratory is located has not been escaped from our facilities.
Right.
So just wanted to point that out every time that we have an audience now to remember that our biosafety, level 3 biosafety facilities are completely safe.
Okay, well, thank you. Right, so before we get started with this session today, I would also like to mention that by the end of the session, we will have some time to answer some of your questions. And to ask a question, just click on the hand button below next to the microphone. You can click the button at any time, and by the end of the session, we will give you permission to ask the question, okay? So feel free to just click on that button whenever you feel that you want to ask a question. Enric, are you ready for your first question?
Let's do it.
Okay. So, Enric, you've been closely involved in advising the Catalan government on lumpy skin disease, and your team is actively working on this emerging virus. To help our audience understand the bigger picture here, could you tell us about the main focus of your research team regarding lumpy skin disease?
Yeah, sure. So the thing here is that lumpy skin is the first time that has been diagnosed in Catalonia or Spain. No? So we were previously a disease-free country. So for all of us, it's been new disease. Yes. So a disease that also it has a peculiarity. It's characterized as a category A disease by the EU legislation, no? And that means that the only option that we have here is to do an immediate eradication. This implies measures that are dramatic, no, for the sector, including stamping out of herds with the economic and emotional consequences that this has. So we were, as you said, asked to settle a committee to help the the
Right.
management of the outbreak. But one of our first concerns was to learn about the disease. We are experts in animal health. Yeah, we've been dealing with African swine fever in our lab, tuberculosis, prion diseases, but this is a new virus, no? So our first goal, in a way, was to get to know the disease. To do that, we went to these camping out scenarios to collect samples, perform a few necropsies, be able to have the virus ourselves isolated for future studies. No, we want to sequence the virus and try to compare its sequence with other outbreaks in Europe, and maybe try to find out what is the origin. No, this is the first question that everyone asks when an outbreak like this occurs. How has it arrived? We still don't have this answer, nor
Right.
Italy or France. No, the previous countries in Europe where outbreaks more recently appeared, they don't know yet how the virus arrived to their territory. We can later maybe hypothesize. No, this is because of animal movements. It's because of vector movements. Um, we don't know. So that was the first approach we did. With that, we gathered samples, we gathered tissues. We started studying, for example, the evolution of lesions, of the skin lesions, to understand where there is the virus. To do so, we have set up in situ hybridization technique that allows us to locate the virus within the lesions and through different stages. And this is now slowly giving us more knowledge on this disease. Um, Also, of course, we have a very strong epidemiological line in CReSA, no? And these people were obviously included in the scientific committee, and they have been trying to trace the movements of the animals and the network contacts to try again to explain, no, how this disease came to our country. The truth is that we haven't been able to demonstrate the origin yet.
Okay.
One of the most plausible explanations is that a movement of live animals from France, our neighbor country, must have brought the virus from there here. No, because there's free commerce, live animals, and each week in huge amounts, thousands to our country. No, and if a region is free of lumpy skin, these movements are legal. So at one point, a movement like this might have happened, and then because the disease spreads through vectors, hematophagus vectors bite an infected animal, and in this way, they say apparently it's a mechanical transmission. So the virus present in the mouth of the insect is then inoculated in the next animal, and that is the way that the disease spreads in the territory. No, more or less, they say that about a kilometer per day can be this the speed at which the disease spreads. And yeah, so probably a movement that we have not controlled brought an infected animal. This animal infected some of the farms in the region of Alampurda, which where the initial outbreak was detected, and from there it spread. But we haven't located that specific movement. No, all the farms have been analyzed so far, and none of them have a specific movement from France, from a region that later became positive or so. We have analyzed all the movements in the few months before that. Another thing that we've done is to study the age of the lesions, in each farm, to try and establish how long was this farm infected. The first outbreak, for example, the one that the farmer or the veterinarian that raised his hand and said, "Hey, I think I have a disease in my." This wasn't the first case. The lesions in this farm were very mild, very acute, and there was only a small percentage of animals with lesions, lower than 2%. Right? So we know that this wasn't the first. Later, we find other farms with higher percentages, around 35 or even higher percentages of animals with lesions. And some of these lesions were in more advanced stages. No, the disease, what provokes is a nodular dermatitis, no, presence of nodules. These nodules are infarctions. No? The disease also provides causes vasculitis, and this vasculitis provides infarctions areas in the skin that then the animal reacts to this dead portion of skin and expels it. No, there's the formation of a crust. In English, I think they call it a sit fast, which, by the way, contains viable virus. So it's one other mode of transmission within the farm, not as important as the vector one, apparently. I lost my train of thoughts here. So, yeah. So what we established is for each farm, how many weeks had been infected, no? And this gave us the information that for 2 months, at least since the beginning of August, the virus had already been circulating in Catalonia, no? And
All
these
right.
months have allowed it to spread to as much as I think we have now 18 confirmed outbreaks. We have had. At the moment, there is no active outbreak.
So this would be like the main results from this preliminary research that you
Yes,
carried
exactly,
out.
exactly.
Okay.
This is what the first reaction to the outbreak. No, try to understand it, try to get to know the disease.
Yeah.
My discipline is pathology, so the first thing I needed is to adapt a necropsy. No, to I want to see the disease. Also, we'll talk about control measures later, but there is obviously an emergency vaccination in place, no, for this disease, and we were also asked to perform vaccine response studies because it's always good, no, even though these vaccines have been produced and there's been studies before their approval and everything. We know they are effective. They've been used in other contexts, you know, like the outbreaks in around 2015 in the Balkans. But it's always good to evaluate how are they working in our context. No, every context is different. So
Yeah.
what we did is we started a couple of vaccination response studies. It was a coincidence, but one of IRTA's farm was in the restriction zone, so it was of the one of the first batches of farms that were vaccinated. We were lucky because in just a week from the declaration of the first outbreak, the ministry, the agriculture ministry, had already acquired vaccines, and in just a week we were vaccinating. So hopefully this will have helped to contain the outbreak. No. So what we did, yeah, in this farm and another farm, because we wanted to test two of the different vaccines that were being distributed, we studied the the vaccine response. No, we analyzed the temperature, the milk production in one of the farms, viremia. It's an attenuated vaccine, so it replicates in the animals. So we wanted to check if they had viremia, excretion of the vaccine virus through oral and nasal secretions, antibody response, and then cellular immunity response. So what we saw, if you want, I'm gonna make a little summary of the results. No, it's maybe not
Sure.
the goal today to explain these studies, but basically we have a low percentage of seroconversion. It is around 35% of the animals in both farms seroconvert,
Okay.
and but on the other hand, when we look at the immunity, cellular immunity responses, all of the animals in one case in the OBP vaccine, all of the animals at some point they have cellular immunity response. No, the test we did was the gamma interferon liberation assay, and not all of them at the same time, but in the different points we analyzed, all the animals responded. In the other study, however, a few animals, 4 to 5 animals, didn't respond in the time the study lasted, didn't have a clear response. But all in all, the functioning of the vaccine seems okay.
So you think that to control the endemic areas, vaccination is enough?
Well, not.
Well, apart from biosecurity, of course.
Exactly.
But vaccination is like a very important strategy to implement and completely necessary.
That's exactly it. So eradication is possible because we've seen other countries do it, but it is not possible without vaccination. No,
Okay,
but
I have a question.
the main strategy that's being carried out now is the stamping out. So with the stamping out, it's a harsh measure with lots of emotional and economic costs, but it's the measure who most efficiently eliminates the virus and
Yeah.
prevents its spread. No, and what is the situation with this disease? Is that the response, the individual response of each animal is very different. No, and we have a high percentage of animals that see the virus but won't develop the disease. But there's studies that show that these animals can be infectious. And that's a huge risk. No? If we don't do the stamping out, if we don't do the vaccination, even if we kill the animals with clinical disease, with apparent skin nodules, those subclinically affected animals will be infected. And if this animal travels to an area without vaccination, for example, also a risk that the virus then moves to that direction.
So
And
I have a
obviously,
question regarding vaccination and stuff. So lumpy skin disease is very, very close to sheeppox and goatpox viruses, and we still do not have a vaccine for these diseases. Do you think it makes sense for pharmaceutical companies to maybe develop a vaccine that could be protective of these 3 diseases and be administered to these 3 species?
I mean, obviously, if that were a possibility, it would be great. Yes. So I encourage companies to do research in this sense. Actually, live attenuated vaccines for sheeppox and goatpox have been used, and they do
Yeah.
confer certain protection to lumpy skin disease virus, which is obviously another capipox virus, but it's not as efficient as the homologous vaccine. No, they are candidates that should be considered because we might face situations where there is no availability of vaccines for lumpy skin. And in this case, I would rather recommend use heterologous vaccines because at least you'll have some degree of
Partial
protection.
protection.
Exactly. But even though the serological responses are indistinguishable between these 3 viruses, the cellular immunity probably has certain differences that, well, I don't know, maybe there could be a strain that matches the 3 of them.
Well, let's see. Maybe it's like an area that could potentially be explored in research.
Exactly.
Right?
Exactly.
Okay. So you mentioned some complexities when studying lumpy skin disease under lab conditions, because as you said, not all animals get infected or develop clinical signs. So maybe sometimes it's quite difficult for pharmaceutical companies to assess the efficacy of those vaccines, right? So how does this variability affect the way when we design experiments? Because, for instance, understanding that there's this variability, you might increase the sample size, but sometimes in cattle experiments, it's quite difficult to just include more and more animals. So what are your thoughts on that?
Yes, exactly. That clinical variability is a difficulty when doing studies. You are totally right to say that increasing cattle studies, cattle numbers is an issue, and specifically if you are working in a level 3 biocontainment level, because then our boxes have a very limited capacity. Yeah, it is a challenge. Obviously, there are ways to overcome this. For example, in the way you inoculate the virus, right? The natural way would be to do an intradermal inoculation. No, that's to stimulate an insect bite, where it delivers the virus in the dermis, and then this virus is picked up by the dermis monocytes, and from here goes to the lymph nodes, and then produces a viremia. No, so one approach that in the we have maybe we can talk about this later. We have asked for funding for a research project, and we have proposed this, no, and it's been done in other experiments, is to do an additional intravenous inoculation of the virus. No,
Okay.
in this sense, you overcome this possible contention of the infection. It's a less natural approach, of course, but in the end, it depends on what responses are you trying to obtain. No, so
Yeah.
yes, there are ways to overcome a little bit this challenge.
And in your vaccination response studies that you already carried out, could you solely rely on nasal shedding or saliva shedding to assess the excretion of this virus, or how would you manage to assess the transmission of this virus through other animals?
No, well, so it is known that the virus is excreted in eye or ocular secretions, nasal and oral secretions. And actually, for the diagnosis of the disease, these are some of the samples of choice, as well as the PCR in blood, no, to test the viremia. But also, even the viremia in these animals, it's not constant. It may happen just a few days during the course of the disease. So, from a diagnostic perspective, the ideal sample is always the skin lesion. This is where the virus is located. Okay. Of course, in our what we when we are thinking of a bioassay, and then we have access to all the samples, we will do even if no lesions appear, we will do skin biopsies. No, not only because of this variability in the viremia, but also because the skin is where the insects capture the virus from. So it is of interest to know if we can locate the virus there. So yes, we have planned some bioassays, and these bioassays include, apart from oral, nasal, and ocular swabs, include skin biopsies. Even if there's no nodules, no, we're gonna go to these lesions like the head, the neck, where lesions are more prone to appear, and make sure there's no viruses there.
Okay. Now this is more field application question, maybe. Do you think it is a possibility that lumpy skin disease will be endemic in Spain now that it has arrived? Or do you think we are close to eradication?
So this is the crystal ball question, no? What's going to happen in the future? First of all, let's try to be optimistic. Eradication is possible. That's why it is that's why the disease is classified as category A, because there are certain free countries, and we have the tools to eradicate. We have stamping out, we have vaccination. Every day we are more aware of biosecurity measures. Some industries, like the pig industry, is way ahead of others regarding biosecurity. So at some point, the bovine industry will also have to try to use these approaches, no?
Yeah.
And we've seen it in the Balkans. They've had an initial outbreak in 2015. They started vaccinating, and then the winter came, no? And from January 2016 till April, they had no cases. So they were, okay, we did well. It's a little bit the same situation that we are now, no? But with the difference that we already had the cases in Aragon, and one case in January in Catalonia. But right now, we have no outbreaks. We haven't had them for a while. The restriction zone is already lifted, so movements are happening again. The farms that were stamping out, they're already bringing animals in. These animals are being tested, and so far they are negative, so good news. But what happens? now the spring will come, temperatures will rise, vectors will start doing their things, and there's a risk. There's a risk that happens again. What happened in Greece? In April, they started having loads of outbreaks, and the disease spread to all the Balkan countries. And all those areas that didn't have a good herd immunity kept reporting cases. When the vaccination coverage was already good, Greece and other countries stopped having cases, and in 2018, only cases were seen in Albania, which was the region with the less percentage of vaccination. And finally, 2019, they vaccinated again, and no more cases appeared. Okay, in 2020, they stopped vaccination. So I think if even though the production systems in those countries and ours are probably different, no, we produce in a more intensive way, bigger farms probably, more pastures. We have lots of animals going to the Pyrenees, etc., which is also it's a difference, but it is also it makes it difficult to manage, no? Especially in maintaining this vaccination coverage. For example, they spend many months in there, no? So calves that have been born now and they have to go up to the pastures, those calves have maternal immunity for a few months, but they will lose it. And if they lose it once they're on the mountain, it's logistically really complicated to gather them and vaccinate them, you know? So this is going to be an issue because when these animals lose their immunity, if they have not vaccinated, there's a risk that they are infected. No, if the virus is still in circulation because of the vectors. The vectors.
Yeah.
So this is why also it is very important when you do the vaccination, the emergency vaccination. We must remember it's an emergency vaccination. That has to be authorized by the authorities. None of the vaccines we are using are actually registered in Spain.
That's right.
They have been authorized exceptionally to contain this outbreak. So when the vaccination is done, it's very important that it is accompanied by a very thorough clinical examination. And by clinical examination, I mean that the animals need to be palpated. You have to touch the sides of the animals, the legs, and make sure there's no nodules, because some of the breeds that have really a little bit of long hair, they will hide these nodules. And when you see nodules, these have to be biopsied. No, just an oral swab and a PCR in the blood might not be sufficient. So yes, one of my recommendations here is to try to detect the outbreaks as early as possible once we know the virus is here. And this, of course, will mean that you will make many biopsies that are going to be demodicosis, or are going to be bisnoitiasis, or are going to be other, an insect bite can cause nodules like that, no? And it is an effort, but early detection is the most important rule. So we might be in a situation like Greece that in the next spring, more cases appear. We cannot rule out that. Okay, because some of the herds that we're vaccinating already had the virus will be still circulating. Whether it will be endemic or not, as I said, we know it can be done, but it also happened again in Italy. It happened in France. It's spreading in France. So we cannot deny that there's a possibility that this will end up being an endemic virus. We are not in this framework right now. We're working on the assumption that we can eradicate it. But yes, it is possible. It is possible. And in that case, it would be advisable for companies to think of producing and registering a vaccine so that as a country we can be self-sufficient and not depend on vaccines produced elsewhere. No, the European Union has a reserve of vaccines for cases like this and to help contain the disease. But if the disease spreads to many countries, it won't be enough. So it's maybe on the safe side to start thinking maybe we should have our own vaccine.
So if we became endemic, hypothetically, what would be like the control strategies to adapt? Because of course we cannot like continue just euthanizing animals that are infected or limiting the the transportation and stuff. So how do we adapt to this situation?
Well, again, this is a hypothetical
Yeah,
case.
it is. It
The
is.
framework now is to eradicate and stop the dispersion of the disease. Once we're endemic, obviously the stamping out makes no sense because it's too expensive, no? In that case, then we'll have to go to a scenario like blue tongue and add the lumpy skin vaccine as another routine in our herds, and also we'll have to make an effort to improve biosecurity. No, this is
Okay.
it's becoming more and more important. No, for animal health, treatment should be the last option or even not consider it. No, we have to always prevent, either by management, biosecurity, and if needed, vaccination. The problem with that is that once you start vaccination from monkey skin, you cannot be considered as a disease-free country, and this has
Exactly.
consequences. So I think the European Commission is now observing this scenario in which we can and we should eradicate, and not in the becoming endemic. But it's a risk. It's a risk that it's on the table.
You were encouraging pharmaceutical companies to maybe develop their own vaccine. So I have also a question about that, because would you encourage to produce more a vaccine, like to spend more money on vaccines, or maybe find like other novel treatments such as antivirals or other treatments that could prevent lumpy skin disease? Understanding that the vaccine, maybe we have non-responders, and well, maybe they produce like a better vaccine or with better immunological response or whatsoever. What are your thoughts on that?
Yeah, no, as I just said, I think that the antiviral treatment, unless you find a really good one that is very effective and cheap to produce,
Yeah.
I don't think it's the option, no, for
It's
especially for
viable. Yeah.
livestock. I think prevention is always the way to go.
Okay.
The vaccines that already exist, they are very effective. It's true that it's a live vaccine, so its stability it's limited. It
Yeah.
only lasts for certain hours, but it provides a very good immune response. So maybe it would be a political aspect of it, no, to be able to have our own vaccines and be self sufficient, not depending on others. Obviously, vaccines can always make them better. Other ways to deliver the antigens that confer better protection and have less side effects. This vaccine is quite safe, but there are local reactions in the inoculation point, the neck.
Maybe
Some of
fever.
them, huh?
Maybe fever the few first
Yeah,
days.
it is described that there is fever, there's a few loss. There are random descriptions during these months that, oh, I vaccinated the cow and it died immediately. Well, an anaphylactic reaction is rare, but might happen. But
Yeah.
it's very seldom, no. But it is described in these cases is that at least 1%, and this is not minor because there's loads of animals, might develop what is called a Neethling reaction, no? And it's the appearance of nodules, not just in the neck, but also in other parts of the body. And what is really important in these cases is that this must be diagnosed. These nodules must be biopsied and sent to the reference lab, and there is a PCR that will distinguish between the viral strain and the field strain. No, and failing to do so is again a very dangerous thing for the remaining
Yeah.
animals or the remaining farms. No, because if the neighbor farm, if this is not tested and it turns out to be the field strain, and it's because you have the rest of animals vaccinated, you won't see more clinical signs in this. If you do not test this, and then the farm next to you has a few calves that have lost their maternal immunity, but because they're only 3 or 4, you still cannot vaccinate them because the vial is used for
Yeah.
20 animals. So logistic things like that may end up in a new outbreak. We have,
Okay.
for example, I didn't comment this before, but one of the latest outbreaks that we had, it was in some pastures, in which at the moment of vaccination, it was impossible to collect all animals. So a few animals stayed up. And those animals, three of those animals showed clinics. No, so this is to stress the importance of keeping that levels of vaccination high, because of these few animals that were not vaccinated, that became an outbreak. They had to do the stamping out with all the horrible consequences that this has, and then movements were restricted for 28 more days in that region, which also is economic losses for everyone that cannot sell their animals or enter new animals. No, so at this point, with these measures, in fact, it's worse. The legislation about the disease is worse than the disease itself. No, now, but we have to look at the bigger picture, and right now this is the situation.
Yeah, I mean, in the end, we need to think like globally and not just on our own, because our own actions can like affect our neighbors, our fellows. So it's really important that we think as just one.
Exactly. And there is an issue that is under discussion, and it's the possibility of doing partial stamping out. That is only sacrificing the positive animals, right?
Okay.
That, of course, is something the farmers would agree on, no? Because losing all the farm is really harsh. But there is we've already talked about this. There is an inherent risk because of asymptomatic animals. And even if you did the PCR test on blood, the PCR, the viremia is not continuous. So there's some animals that you won't diagnose. No, it is a good tool, the PCR in blood, for to diagnose a herd, because if you sample enough animals and you have the virus, it will come up. But to leave subclinical animals and PCR negative animals in a harm, it's a risk that right now, at least the commission is not ready to assume. Maybe in a future scenario where there's multiple cases and it is impossible to pay
Yeah.
for the sacrifice of all these animals, then it would be a measure that would have to be considered. No, it has been used in certain countries, and the regulations. There exists as a possibility, no, but right now it's not authorized.
Well, let's see what the future holds, right? Right. Another question about transmission and shedding. You mentioned that the virus can be shed through the ocular secretions, nasal, and also saliva. But I've read some papers that were also explaining that bovine semen could also be a way of transmitting the disease to cows. Is the semen being tested right now here in Spain?
To my knowledge, I don't think it is. I have no knowledge of that. It is true that artificial insemination is a way of transmitting the disease, but we have to consider that only 5 months ago we were free of this disease. It had never
Of course.
existed, no, in our country. When the outbreak in the Balkans happened, maybe we should have started thinking, oh, should we test sperm? Maybe. How did it come to infect Sardinia and the north of France, this virus? We don't know. Is it through semen imported from somewhere else? Because that might be an option.
Yeah, of course.
There's other hypotheses. No, for example, in the case of Sardinia, we know that culicoides midgets, the ones that transmit blue tongue, have been known to travel from the north of Africa to Sardinia. We talked to our colleagues there, and they don't think this is the case because the cases appeared in the north part of the island, whereas these blue tongue outbreaks have always been in the south. And apparently, there's some mountains in the middle that would prevent the midgets to fly as far. But again, we don't know. So sperm might be, yeah.
Maybe testing the semen, the sperm, could be like a control measure if we ever become endemic. Like in a way of like making sure, a little bit like in PRRS, maybe it's not your area of expertise, but it's routinely being tested so farms can like control a little bit their outbreaks.
That would certainly be a measure. Also, but I think that for example, one of the higher risks now is the movement of live
Yeah.
animals, no? And maybe also testing these animals, which is not, it's not allowed right now because we are in the EU, no? So if you an animal comes from a free region, um, you cannot do these tests. It's illegal.
Yeah.
But maybe these measures should be considered to avoid spreading while the outbreaks are active in certain countries. Yeah.
All right, I have one last question. It's more a global question in this case because you had worked with multiple emerging diseases, transboundary diseases, so you have a unique vision on that. Have you seen like a shift of these emerging diseases? Because in my opinion, I'm sensing that over these last years, there has been more and more emerging diseases here in Spain happening or somewhere in Europe even. And I feel like it just increasing the numbers of diseases that are being brought new to the table here. What are your thoughts on that?
This is a difficult question to answer.
Yeah.
Um, Look at the example of lumpy skin. No, it was first diagnosed in the late 30s in Africa,
Mm-hmm.
and it took it 50 years to reach out of the African continent. No,
Yeah.
obviously, and then it spread rather quickly through all of the Asian countries, Russia, and now it's affecting us. No, and obviously this has to do with the human intervention, the the globalization, the increased means of transport, animals moving here and there. This, of course, has accelerated the spread of this disease, and we cannot ignore the effects of climate change, of course,
Of course.
especially when
Yeah.
we're talking about vector transmitted diseases. The vector populations are constantly changing. We've seen this with the tiger mosquito, for example. No, the tiger mosquito, which we don't know if it transmits lumpy skin, by the way. We know Aedes aegypti is a good transmitter of the disease, but we have no clue. And that is one of the studies that we want to make in our BSL is to do a competence study as a vector of the colony we have of tiger mosquitoes, Aedes albopictus. So yes, it is clear that human intervention and climate change have provoked a change in how diseases. But on the other hand, we also now are way more interconnected, and that helps us knowing in real time what happens, and that is why we are more aware of these things happening. But transboundary diseases have been happening for a long time. Like we had African swine fever
Yeah.
before, and we eradicated it. We had classical swine fever, and we eradicated. And what happens next year? We know that small ruminants and peste de petits ruminants, no, it is affecting Africa as well as foot and mouth disease. So any day this year could happen that the disease is brought from northern Africa to us. So yes, what I think is that we have to be aware of that, have a good preparedness, and work on our biosecurity. More and more, you find you go to an airport and you see it's forbidden to bring pork. But nowadays, I can go to London with some ham in my bag, and we have African swine fever here. So yes, we have to be aware of these measures and yeah, and be careful
Yeah.
because
Yeah, that's right.
new
As you,
scenarios will bring new situations. No, for
yeah,
example,
as we said,
for
like
this,
think globally,
yeah,
not
yeah, yeah.
just
For
on
this
Europe.
reason, in our case, it's important. No, we haven't talked about the project that we want to do, but one of the things we are intending to do is to see how local domestics and wildlife potential reservoirs might be affected by this virus. No, we know that it only affects buffaloes, but if you go to Africa, they have wildlife reservoirs there. They have different types of bovidae, like gazelles, mantengs.
Mm.
There's quite 10 or seven species that have been diagnosed with lumpy skin disease, and also outside of Africa, camels and giraffes. No, So we haven't been exposed previously to this virus. No, it's important to know, or deers or roe deers, fallow deers, might they be susceptible? Apparently not, but there's no experimental evidence. So we will try to do that with red deer and also with sheep and goats, because we don't. It is not clear the role they might play. No, we know they can seroconvert, and that apparently they do not transmit the disease. But again, there's no experimental evidence of this. No, so that's one of the things that we want to try.
Okay. Well, Enric, as we wrap up our conversation, I always like to ask our guest if there is one key message that you would like to tell the audience to remember today's discussion.
Well, yes, the message I would like to transmit here is that we shall remember the context we are in. It's a disease, lumpy skin, a disease that can be eradicated. We have the tools to do it, the stamping out, the movement restriction, the vaccination. But to take into account the importance of early detection, right? I urge every veterinarian and every farmer to inspect their animals, and if they see nodules, report them. The consequence might be devastating, but again, we have to think of the whole thing, no? And not declaring cases now can be fatal and can hinder all the efforts and that we are putting into trying to. And when I say we, I don't mean CReSA, I mean our society, because the sector is very important here. So yes, and if you enter animals from other regions, do a clinical examination when they go, and 28 days after, which is more or less the incubation period in which they can develop lesions, start palpate again your animals and make sure they don't have nodules. And if you see any nodule, confirm it.
Okay. Right. So I don't see that anybody has a question, but I would like to remind the audience that they can click on the hand button below and just we will give you permission to ask the questions. So if there's anyone that has something that they would like to say, now it's the time. If nobody has a question, then I would like to thank you, Enric Vidal, for joining us today and sharing your outstanding expertise on LSD. It's been an insightful and fascinating conversation, and I hope that our listeners enjoyed it as well. Right, so looking forward, our next session will be in April 22nd, featuring Bridget Traveria, an expert in horses' lameness. Okay, so thank you, Enric, for your time.
Thank you very much.
Yeah, hope you had a good time as well.
Yeah.
Okay, see you.
Keep in contact. Bye.
Bye.